Hi everybody, and welcome to our module on purine metabolism. Nucleotides are the building blocks used to assemble DNA and RNA, and there are two subtypes of nucleotides. There are the pyrimidines and there are the purines. The pyrimidines have a single ring in their nitrogenous base, and the purines have two rings in their nitrogenous base. And in this module, we're going to talk about the biochemical synthesis of the purines, adenosine and guanosine. Before we proceed with discussing purine biochemistry, let's talk about some of the roles that nucleotides play inside the cell. So as I just said, they can serve as building blocks for RNA and DNA synthesis. In addition, you can see nucleotides used as a source of energy. You probably know that ATP is the major cellular source of energy, and ATP stands for adenosine triphosphate, and that is the nucleotide adenosine. The levels of cyclic AMP, you may know, lead to increases and decreases in blood flow, and cyclic AMP is cyclic adenosine monophosphate, and that is a nucleotide. Cyclic GMP also serves as a second messenger, and interestingly, it is usually the purines, adenosine and guanosine, that serve as second messengers. We don't often see second messenger systems of pyrimidines like cytosine and thymidine. So let's talk about the sources of the nucleotides that our body uses for all the things we just talked about. Well, you can get nucleotides from your diet. These are called exogenous nucleotides. But then there are also a couple of ways by which the body can synthesize them. These are called endogenous nucleotides. So there are direct synthesis pathways by which the body can assemble nucleotides from building blocks like amino acids and sugar molecules. And then there are also salvage pathways so that when DNA breaks down, the body can salvage the bases and turn it back into nucleotides that it can use for various purposes. So a couple of key points about nucleotide metabolism before we get into the biochemistry. RNA is always synthesized first. Ribonucleic acids are made first by the body, and then the RNA is converted to deoxyribonucleic acid by removing an OH group. That's why it's called deoxyribonucleic acid. In addition, you should know that there are different chemical pathways for purines versus pyrimidines. In this module, we're going to talk about purine synthesis, and there's another module for the pyrimidines. And then finally, you should know that all the nitrogen in the bases of the nucleotides comes from amino acids. There's nitrogen in the air around us, but our body doesn't have a mechanism for incorporating that nitrogen into chemical structures. So when you see a nitrogen group, you should know it comes from an amino acid. And the word amino means that it has an amine group. This is an amine group here, nitrogen and two hydrogens. So amino acids always have these nitrogen groups, and that's what the body uses when it needs nitrogen in a molecule like a nucleotide. So now let's get into the biochemistry of how purines are synthesized by the body, starting with basic structures like amino acids and sugars. So the goal of purine synthesis is to create a molecule of either adenosine monophosphate or guanosine monophosphate. I've drawn the structure of adenosine monophosphate on the bottom here and also guanosine monophosphate. They both have one phosphate moiety, that's why they're called monophosphate. They both have a sugar in the middle. This is a 5-carbon sugar, which is ribose. It's also sometimes called a pentose because it contains 5 carbons. And then they both have a base over here on the side. In this case, it's adenine, and in this case, it's guanine. You can see that adenine has a nitrogen group, and guanine has an oxygen group and a nitrogen group over here. So the ingredients that the body is going to use in order to synthesize this final product of either AMP or GMP is a ribose phosphate sugar, and this comes from the HMP shunt, which I talk about in another module. Certain amino acids will also be used. We'll talk about that in a little bit. And then finally, some carbons will be added. They will come from either tetrahydrofolate or from carbon dioxide. Step 1 in purine synthesis is to create a chemical structure called PRPP. PRPP stands for 5-phosphorybosyl 1-pyrophosphate. It is a structure that's synthesized from ribose 5-phosphate, which comes from the HMP shunt. You can see that ribose 5-phosphate has a phosphate group here. It's got a ribose sugar. That's how it gets its name, ribose 5-phosphate. And what you are doing in this first step of purine synthesis is adding phosphate groups to this OH group right here, and that creates a molecule of 5-phosphorybosyl 1-pyrophosphate, or PRPP, and that's the first step in purine synthesis. Step 2 in purine synthesis is to create a very important molecule called IMP. IMP stands for inosine monophosphate, and this is the chemical structure of IMP on the right side of the screen here. And you may notice that IMP looks very much like a nucleotide, and that's because it is a nucleotide, but it is not a nucleotide that is used in DNA or RNA. It is an intermediate form that is used to synthesize the nucleotides that DNA and RNA like to use. But just like all nucleotides, it has a phosphate group, it has a ribose sugar, and it has a base. And the base that inosine monophosphate has is called hypoxanthine. purine, and we'll talk a little bit more about that in the next few slides. You should also know that in the hypoxanthine base, you can see that there are nitrogen groups and lots of carbon groups, and these all come from amino acids and folate and CO2, and we'll talk more about that in a minute. So let's pause for a minute and talk about the chemical structure of the purine bases. All of the purine bases have two rings with two nitrogen groups in each ring. One of the rings is 6 units in size with 3 double bonds. The other ring is 5 units in size with 2 double bonds. Over here on the right is the structure of a typical purine ring. This one has 6 units for its structure, and this one has 5. And you can see that the 5-unit ring has 2 double bonds, which I'm circling here. And the 6-unit ring has 3 double bonds, the same one from the 5-unit ring, and also 2 more right here and right here. And all 3 of the purine bases have this structure. So this is adenine right here. You can see a 6-member ring and a 5-member ring. This is guanine, and this is hypoxanthine. Most people know that adenine and guanine are purine bases, but don't forget that hypoxanthine is as well. It's just not used in the final DNA or RNA product. It is an intermediate base used to derive these others from its structure. They like you to know the source of the molecules in the purine rings for your boards. And remember what I said before, all nitrogens come from amino acids. So the amino acids that give nitrogen to purines include glutamine, which gives 1 nitrogen to each of the 2 rings. Aspartate gives a nitrogen to the larger 6-member ring. And then finally, glycine contributes all these structures in the middle, the carbons and the nitrogen group as well. The carbon sources are as follows, just like I said before. Glycine gives these 2 carbons right here in the middle. Carbon dioxide gives 1 carbon molecule to the large 6-member ring. And then tetrahydrofolate contributes a carbon group to each ring. And a key point here is that tetrahydrofolate is derived from the vitamin folate, and it contributes to the formation of purines. We'll talk in the pyrimidine module on how folate is very important for making the nucleotide thymidine, but you should also know that folate plays a role in synthesizing the purines as well. Folate is often called a single carbon donor, and that's because just like it's done here, it often donates a single carbon to structures like nucleic acids and amino acids. So here's a summary slide of the key points to remember about purine synthesis. The process begins with a 5-ribose phosphate molecule, which comes from the HMP shunt. The key intermediates are PRPP and inosine monophosphate, and IMP can be converted into the final products of AMP and GMP. And this whole process involves the use of amino acids, like I've shown here, and also tetrahydrofolate and carbon dioxide. There are, of course, many more steps than what I've shown here. You can look these up in a biochemistry textbook if you wish. What I've shown you on the screen are the most important steps to know for step 1 of your boards. You should be aware of how this process is regulated. When the levels of AMP or GMP become elevated, they can inhibit the conversion of IMP. And then the rate-limiting enzyme in this process is an enzyme called glutamine PRPP aminotransferase. This enzyme is inhibited when the levels of IMP, AMP, or GMP become elevated. Everything I've talked about up until this point has been about the synthesis of purine ribonucleotides, but now let's talk about how cells in the body synthesize purine deoxyribonucleotides. And deoxyribonucleotides lack this hydroxyl group here. That's what makes them deoxyribonucleotides, and they are used in DNA. So the way the body synthesizes these molecules is it first synthesizes diphosphate versions of purine nucleotides. So this is adenosine diphosphate with 2 phosphate groups here. This is guanosine diphosphate with 2 phosphate groups here. Once this has occurred, the enzyme ribonucleotide reductase can remove the hydroxyl group, and we then get molecules of deoxyadenosine diphosphate or deoxyguanosine diphosphate. For all the biochemical pathways that you will learn in medical school, you need to focus on the drugs and diseases that are involved in those pathways. And for the purine synthesis steps that we've discussed up to this point, there are 2 drugs that you need to know. The first one is ribavirin, which is an antiviral drug, and it inhibits an enzyme called IMP dehydrogenase. And this is an enzyme that converts IMP to GMP. So ribavirin will inhibit this conversion, and this will blunt the synthesis of guanine nucleotides, which of course are purines. This means there will be fewer guanine nucleotides around for the virus to use when it tries to replicate its genome. There is also an immunosuppressant drug called mycophenolate. It also goes by the name Cellcept, and it inhibits IMP dehydrogenase in white cells. So this will decrease the pool of guanine nucleotides available to T cells and B cells so that they cannot replicate, and this is why it's an effective immunosuppressant. So nucleotides do not last forever. They will break down inside of cells, and free-floating purine bases will be found inside of cells. So let's talk about the potential fates of those free-floating purine bases. So the three purine bases, recall, are adenine, guanine, and hypoxanthine, and they can either be converted to uric acid, which can then be excreted by the kidneys, or they can be remade into nucleotides, and that is called purine salvage. So purine salvage will salvage the bases adenine, guanine, and hypoxanthine and convert them back into the nucleotides AMP, GMP, and IMP. In order to do that, you need that molecule we talked about before called PRPP. Recall that PRPP is phosphoribosyl pyrophosphate, and it is basically a nucleotide with no base over here. Instead, it has two phosphate groups. So what the cell will do is take this molecule and add it to one of the free bases and lop off the two phosphate groups, and then you will have a nucleotide again. There are different enzymes that function for the different purine bases. So for hypoxanthine and guanine, there is an enzyme called HGPRT, which stands for hypoxanthine guanine phosphoribosol transferase. And it should make sense for you, based on this big ugly name here, that this enzyme is going to combine either hypoxanthine or guanine with PRPP to make the nucleotides either IMP in the case of hypoxanthine or guanosine monophosphate in the case of guanine. Adenine has its own enzyme, but it serves the same function. It's called APRT, which stands for adenine phosphoribosol transferase, and it combines adenine with the PRPP, and once you do that, you will have adenosine monophosphate. There's an important clinical correlation of the purine salvage pathway, and that is the chemotherapy drug 6-mercaptopurine. This drug mimics the structure of hypoxanthine and guanine. If you look down here at the bottom of the screen on the right side, I have the chemical structure of 6-mercaptopurine, and it has a sulfur group where hypoxanthine and guanine do not. So this structure right here will be added to PRPP, which I've shown on the screen, by that HGPRT enzyme. The enzyme will pick up the 6MP thinking it is hypoxanthine or guanine when it will not. Once it does this and combines the 6MP with PRPP, it will create a substance called thioinosinic acid. And you can basically think of thioinosinic acid as a mutant nucleotide. It's got the structure of a nucleotide, but it doesn't serve any function inside the cell. So it will get incorporated into multiple steps of purine and DNA synthesis, and as a result, you will have decreased levels of IMP, AMP, and GMP, all because the 6-mercaptopurine mimicked the structure of guanine and hypoxanthine. Another clinical correlation of the purine salvage pathway is the drug azathioprine. It's an immunosuppressant. It's used in inflammatory bowel disease and a number of other disorders, and it is converted inside the body to 6MP, and then it functions just like 6MP. This is the structure of azathioprine at the bottom of the screen. You can see that it looks very similar to 6MP. It just has this extra molecule hanging on inside the body. This molecule will be lopped off, and you will be left with 6MP, and thus azathioprine will exert the same effects as 6-mercaptopurine. So as I mentioned before, purines can be salvaged, but they can also be broken down into uric acid and excreted from the body. So let's talk about how that occurs. So the two purines, hypoxanthine and guanine, are broken down into xanthine and then converted to uric acid for excretion by the kidneys. The enzyme xanthine oxidase will break down hypoxanthine into xanthine, and then it will also convert hypoxanthine into uric acid. For guanine, it has its own enzyme called guanase that converts it to xanthine, but once it becomes xanthine, it is converted to uric acid by the same process as hypoxanthine via the enzyme xanthine oxidase. Adenine has a more complicated process by which it gets converted to hypoxanthine and then to uric acid for excretion. So adenine is first converted back into the nucleotide adenosine monophosphate by that enzyme APRT, which we talked about a few slides ago. Adenosine monophosphate has the phosphate moiety removed, and then it becomes the nucleoside adenosine, which has no phosphate group attached. Then there's a very important enzyme called adenosine deaminase, which removes the amine group from adenosine and converts it into inosine. A deamination reaction is a reaction where you remove an amine group, and that's what adenosine deaminase does. The reason adenosine deaminase is an important enzyme is because it's deficient or dysfunctional in many cases of severe combined immunodeficiency syndrome. I talk about this in the immunology modules, but it's a very severe immunodeficiency syndrome, and many of these cases are caused by dysfunction of the adenosine deaminase enzyme. Once inosine is present, it can be converted into hypoxanthine by an enzyme called purine nucleoside phosphorylase, and then the hypoxanthine can go to uric acid and be excreted in the kidneys by the same process I discussed on the last slide. There's a relatively common clinical condition that ties in nicely with the purine salvage and breakdown pathways, and that is the clinical condition of gout. Gout occurs when there is excess uric acid present in the plasma. Uric acid crystals will deposit in joints, leading to pain, swelling, and redness. The most common location is the base of the big toe, as is shown in this picture right here. Now, this can occur from under-excretion of uric acid. For example, patients who are placed on diuretics like furosemide often under-excrete uric acid, and they can have gout attacks because of this. But you can also develop a gout attack from overproduction of uric acid via the purine pathways. If there is high cell turnover, for example, in patients who have trauma or who have lots of cells dying from chemotherapy, there will be lots of purines spilled into the plasma. Those purines will get converted to hypoxanthine, and then they will be converted to uric acid, and thus you will have high levels of uric acid, and this will be a setup for gout. Another classic trigger for a gout attack is consumption of purine-rich foods. Purines are found in meats and seafoods. You can basically think of anything that has a lot of protein as also having a lot of purines. So if you eat a lot of meat or seafood, you will then have a lot of hypoxanthine in your blood, and this will get converted to uric acid, and the uric acid can lead to gout. A classic clinical scenario is a patient who goes out for a big steak dinner and the next morning has swelling and pain in their big toe. They have a gout attack, and the reason is because they consumed lots of purines at the steak restaurant. The treatment for gout is to administer an inhibitor of xanthine oxidase called allopurinol. This will blunt the conversion of hypoxanthine to uric acid and lower the uric acid levels and make gout attacks less likely. A few slides ago, I talked about the two drugs, azathioprine and 6-mercaptopurine, which mimic the structure of purines. And just like purines, these two drugs are also metabolized by xanthine oxidase. 6-MP is converted by xanthine oxidase into thiouric acid, which is an inactive metabolite. This means you have to be very careful if you co-administer either of these two drugs along with allopurinol. Allopurinol will inhibit xanthine oxidase, and this will boost the effects of 6-MP. Sometimes this is done on purpose to boost the effects of one of these drugs. However, you also have to be careful because co-administration of allopurinol may increase the toxicity because you are blunting the metabolism of the two drugs. A final clinical tie-in with the purine salvage pathways is a rare disorder called Lesch-Nyhan syndrome. This is an X-linked disorder where children are born with absence of the HGPRT enzyme. Recall that HGPRT is important for salvaging the purines hypoxanthine and guanine. So if you do not have HGPRT, you cannot salvage those purines, and thus they will get converted to uric acid. Therefore, these children have excess uric acid in their plasma, and this condition is sometimes called juvenile gout. In addition, because you cannot salvage purines, the de novo purine synthesis pathway ramps up. So these children have very high levels of PRPP and IMP in their systems. There's also a neurologic impairment that goes along with this disease. The mechanism is unclear. It's hypothesized that the high levels of uric acid lead to the neuro impairment, but this has never been shown with certainty. The symptoms of the neuro involvement are hypotonia and chorea, which is a movement disorder. Another classic feature of the neuro impairment is self-mutilating behavior. These children often bite themselves or scratch themselves, and there is no treatment. So the classic presentation of Lesch-Nyhan syndrome is a male child, because it's X-linked, who has motor symptoms and self-mutilation and gout. And they will often ask you about the absence of the HGPRT enzyme or the high levels of purine synthesis via the de novo pathway. So I'll finish with a summary slide to try to tie together everything we've talked about in regards to purine metabolism. De novo synthesis begins with the ribose 5-phosphate molecule, which gets converted to PRPP and then to IMP. IMP can be converted either into adenosine monophosphate or guanosine monophosphate. When you have free purines floating around like guanine or hypoxanthine or adenine, they can be salvaged via the purine salvage pathway. HGPRT can convert guanine or hypoxanthine back into IMP or GMP, and then APRT is what converts adenine back into adenosine monophosphate via that complicated process I showed you a few slides ago. Hypoxanthine and guanine can be converted into xanthine, and that can be converted into uric acid, and then uric acid is excreted in the kidneys. And that concludes our module on purine metabolism.